All right, so at the beginning of last lecture, we had talked about the cell membrane, and we had said that we have these little openings. We had said that semi-permeable membrane, that differentially permeable membrane allowed certain stuff to pass while blocking other things. And we had said, why is that, or how does that work? And we had said, well, a phospholipid bilayer pretty much blocks everything. So we have these other structures. We'd said we have channel proteins, and we have these transport proteins. And so we'll talk about the difference. We're going to explore the difference between these two right now. So when we talked about the channel proteins, I said it's like a straw that allows something in particular to cross. And I said the cell doesn't make it move in or out of the cell. So it's not the protein. The protein doesn't make this happen. It just does. And so since the cell, spit it out, Steve, since the cell doesn't use energy to make this happen, it's a natural process. And so we need to figure out why this passive movement occurs. It's on the board. I've written it on the board. Okay, so I've got a couple of things up there. Let's look at that top one first. It's something called Brownian motion. And you can see, if you can read my writing, you can see it is molecular motion due to either heat or light energy, causing the molecules to vibrate. Okay, so they're, depending on how much heat or light there is, the molecules are vibrating. And so if you're vibrating and you're with a whole bunch of other molecules, then you have this process here called diffusion. The movement of molecules from an area of high concentration to an area of low concentration. If you think of a perfume bottle, you pop the top, and in the bathroom, and at first you smell it, you say, yeah, I can smell it, and you put it down over there, and then the other side of the room, you can smell it over there. And what's happened is those molecules have vibrated into each other and bounced apart in a process we call diffusion, which is a movement from an area of high concentration to an area of low concentrations. And I wrote up here, why does this happen? The vibrating molecules are bouncing apart. Okay, so when we talk about diffusion, we can talk about simple diffusion. And in anatomy and physiology, there are cases where this happens. The movement of blood gases from the air into your circulatory system, into your cells, is driven by simple diffusion. Now, I talked about those phospholipids blocking most everything. They do not block oxygen or CO2. Blood gases, these tiny molecules, they just pass right through. They don't go through a channel. They just go through any of it. Any portion of it, they pass right in or out. And they're driven by the concentration. Diffusion, simple diffusion. It's just you put a sugar cube into a cup of hot water, and you can stare at it so fascinated, and it will disappear. And if you take a sip of the top of that water, the sugar cube was at the bottom, it's sweet. It's because it's diffused. It's filled the space. Now, we had said many things can't do this, right? We had said that they require a straw to pass through, a channel to pass through. So if you are the kind of molecule like water or ions that can't pass through that phospholipid bilayer, you can only enter or leave by going through a channel. And so that is what we call facilitated diffusion. So if you don't need a channel, it's simple diffusion. If you need a channel, it is facilitated diffusion. We have two more things we're going to talk about, but I'm going to, osmosis and filtration. What we have basically in this slide that I'm talking about are passive processes. And by definition, these are processes that don't require the cell to add energy. Yes, there's heat and light energy, of course. But there is no energy from the cell to make it happen. And so we have four different things: simple diffusion, facilitated diffusion, osmosis, and filtration. We're talking about the first two right now. Simple diffusion, we've already talked about it. Facilitated diffusion, we've started talking about it. So if we go to the slide for facilitated diffusion, we had said it involves some kind of a protein, this channel protein. And it's driven by the difference in the concentration. What did we say diffusion is? Movement from an area of high concentration to an area of lower concentration. We said with facilitated diffusion, you have to have that hole, you have to have that straw, you have to have that channel protein. So it's limited by the number of channels that are available. And so there are lots of things that will enter and leave the cell through channels driven by their concentration gradient. What is that? It's the difference in concentration from here to here, between two locations. The difference in concentration between two locations is a concentration gradient. All right, we're going to talk next about a very special kind of facilitated diffusion. It's called osmosis, something to look forward to.